I'm Allison Jonjak, the Cranberry Outreach Specialist with the University of Wisconsin-Madison Division of Extension. These videos are the on-demand recordings of UW Extension and the Wisconsin State Cranberry Growers Association's virtual brown bag lunch series. These monthly sessions offer growers and researchers a way to share timely and relevant information. We hope you'll enjoy and continue to engage with us.
Hi, I'm Tom Lochner, the Executive Director of the Wisconsin State Cranberry Growers Association. We're excited to be a partner with Extension to bring you research-backed resources, tools, and tips to keep Wisconsin marshes productive and profitable.
And so as we kick off, as the growing season gets underway, we are going to have growing season updates. We're going to have today the standard housekeeping. So if you have a question, either unmute yourself to ask a question at any time or type a question in the chat. If you have a question that you'd like me to ask on your behalf, in the chat you can say to and make the chat just to me, and I will ask that question for you anonymously. So we do want people to feel comfortable to ask questions, whether out loud or by chat. We have the live transcripts on for those who like transcripts. Click the more and say view transcript. And this morning, Tom and Alex are not able to join, so they had sent over a quick welcome that I'll give on their behalf. Then we're going to have our consultant update and what we're seeing in the field and what we're looking at in the future with Suzanne Arendt of Red Forest IPM. From DATCAP, Alyssa Foss is going to give us the common questions and common issues with worker protection questions as we move into the beginning of the season. And then Shawn Steffan is going to join us for an overview of how the commercialization of the Nematode projects is progressing, how those patents are working and all of these things. So I'm going to read the quick welcome from Tom, and then I will hand it over to Suzanne. Tom says, here we go. I usually say good morning. Tom usually says good afternoon from the Wisconsin State Cranberry Growers Association. We're sorry that we're not able to join you all today for the May brown bag. Recent activities at the association include efforts to fill the publication, education, and communications manager position and work with the 14-mile watershed group. So those in the 14-mile watershed will wind up hearing more on those progresses. We've also been busy preparing for upcoming events. The Wycliffe Cranberry Open will be held on June 7th at the Bullseye Golf Club. Applications for the Wisconsin Cranberry Leadership Development Program are due by July 20th. The WSCGA annual summer meeting, field day, and trade show will be hosted by the Wisconsin Cranberry Research Station at the station in Millston on August 10th. So mark those dates, and please watch the WSCGA news and your email for more information on those events. And then I will move on to Shawn Steffan. Yes, it looks like you've got a field day.
Yes, so I'm in cranberry country. We are establishing bee nursery posts today, day 2 of a big push. So yeah, I am dialing in from outside of the Culvers. So I'll apologize for any loud engines or sirens going by. Yeah. Okay, well, so I will also try to keep this short and sweet, and I'll take questions as needed. So the nematode propagation and bioinsecticide development is continuing. I had a really good conversation recently with 2 reps. So, so I had this, I'm not sure where you lost me, but I had a good conversation. Okay, great. Yeah, so their methods of nematode propagation are much different than ours. We use live insects, and in so doing, we ensure that our nematodes, they can actually kill an insect. They have to find the mealworm larva, they have to get in and then kill it using their bacteria. And so what that ensures is that the nematodes that we get out, downstream, those nematodes are killers. And they're also good at finding an insect and subduing it. It's sort of like they overcome the immune response of the insect. So because we propagate our nematodes using live insects, I think what we get, out, when we harvest those nematodes is high quality killers. Whereas the industrial scale propagation methods of most of these commercial companies, what they do is they kind of make a big milkshake of sort of beef or lamb byproducts, and so it's meat, and they make sort of like a big frothy slurry of beef smoothie, and then they soak that into a substrate with lots of places for nematodes to hang out. Typically, it's like a big, lots of little cubes of sponges, and then those sponges are like a place to like live and reproduce. Have your daily raw beef smoothie, whatever it happens to be, and then you propagate. You through the generations, you end up with lots of nematodes, and then you kind of rinse these sponges, and you thereby like harvest the nematodes, which is why the Koppert product, which we're testing, their suggested release rate in the field is 1.5 billion per acre, whereas ours are 12.5 million. So they are, orders of magnitude higher concentrations, because the nematodes that they produce, in all likelihood, are not good hunters and killers. But all that matters in the end is that you get good efficacy. You get your nematode into the soil and then onto the target host. And if it takes 1,000 nematodes to kill that target host, so be it. Whereas with our method, I think if we get 1 or 2 in or near that host, our guys kill it. All that matters in the end is that it's cost effective and that it actually kills the insect host. So what I told them is I really don't care about the particular propagation technique as long as we're able to maintain quality control and it's a good price. So the way we left it is they're very interested. They want to talk to me further and USDA lawyers about intellectual property, and then in the near term, we have trials running, 2 trials running up in northern Wisconsin right now. One is the timing trial we set up last September, and the timing was a late summer timing for cranberry fruitworm, Sparganothus, and flea beetle. And so we used our nematodes, our 2 species, with a head-to-head trial against the, I shouldn't say against, but in comparison to the Koppert nematodes. And so we've got to control our nematodes, the Koppert product, and so this comparison will be a really good head-to-head test as to whether like our particular nematode at our rate, their nematode product at their rate, how does it compare? Hopefully both do really well, and it'll be some indication that nematodes applied late summer may be the best timing for cranberry fruitworm, because at that point the larvae have finished feeding in the fruit, they leave and they sort of go into the duff and they spin up in the duff, and there's a period of time when they exist as larvae and not yet pupae, and I think that's the vulnerable time to hit them, when they are larvae spun up in their hibernaculum, which is kind of like a sleeping bag for winter. That timing, that late summer timing, should be great for cranberry fruitworm, and we'll also pick up flea beetles that so basically eggs that were laid in July and August, they may have hatched, so they likely exist as larvae in the soil and sort of in the very shallow parts of the soil, so they may be vulnerable to the nematodes. And then obviously Sparganothus, late summer, they start to go down into the duff to spend winter. So we might be able to pick up all 3, but the main one we're going after for that timing is cranberry fruitworm. So Koppert really wants to see those data, because those data will basically say their product works similarly to ours, or perhaps our product works better, perhaps their product works better. It'll give some relative comparison of the two nematode products to see like whether they should bother going through basically like product development with our nematodes. Like if their nematodes perform really well and ours do not, they will have no interest in talking further about our nematodes. Conversely, if our nematodes do really well and theirs don't, then they'll say, hey, maybe the cranberry market and the blueberry market is going to be good enough in North America. Maybe we should talk further. So that's where that stands. Yeah,
I have
go
a
ahead.
couple of questions in the chat if I can relate them to you. First is, can nematodes reproduce in the field, or is the application the only way that we're really getting these kind of mass amounts of them?
Right. They absolutely can reproduce in the field. They are native animals. We found them in the central Wisconsin peatlands. They are wild animals. They're like, native daughters of Wisconsin. They like Wisconsin winters. They like wet acidic soil, and they like to eat insects that attack cranberries. So the one thing they need, though, if they're going to continue to reproduce in a grower's bed, is food, and food is insects. And so typically there are insects in the soil, in the duff layer, that will represent food for the nematodes. The thing is, like we, crop managers and we pest control people, we work really hard to keep insect numbers down. So we're kind of eliminating their food source. And what that does is it will make the nematodes very spotty in any given bed. And that probably will necessitate augmentation or, broad application. Does that make sense?
Yeah, and I have another question in that bucket, and then I have a second question from another person. Can nematodes survive a diazinon application? If we make our nematode application and then we're controlling flea beetles,
right.
have we upset ourselves?
Probably not. So the nematodes are going to be in the soil, so they should be protected from any residue that's in the canopy, and then any residue that gets washed down onto the soil layers. What tends to happen is they get bound up in the surface waxes of leaves and other plant material. So the only time I think it could be a problem is if you apply the diazinon like the same day or maybe the day before, and there was lots of residue on the canopy surface, and then you applied nematodes, really soon thereafter, and before the nematodes could get washed down into the soil, if they got a hit from diazinon, that might be the only time that you'd have an issue with diazinon. Like basically, if you recently applied it right before you were putting the nematodes on.
Okay, that is good information, and hopefully hearing that will help people avoid that. So it sounds like give it a day or two before making a nematode application after a diazinon application.
Yes. Yeah.
Cool. The next question, and I'm going to actually ask you to just clarify real straightforward. So is Koppert currently testing their own species of nematodes? So they're testing something other than the Oscheius onirici and other than the Heterorhabditis georgiana. Is that true? Okay. And do you have the species name that they're testing?
Yes. Yeah. So I'm actually doing the testing. They sent me lots of product. I mean, tons of product, and which is great because they want they need third-party impartial testing, and it's also nice because they're in one of our trials. It's head-to-head with our own nematodes. The species that they use, that they recommend for, they thought would be best for my our cranberry system is Steinernema carpocapsae. Okay. And their particular product is called capsanem. So it's CAPSANEM. It's like capsanem. Okay. Like carpocapsae, kind of capsanem. And that's their product. So you can go to the Koppert website and you can look at their various nematode products. And the one that we're testing is capsanem, and that is a head-to-head trial up in northern Wisconsin with our 2 nematodes and then a control.
Awesome. I have one more personal question, then we'll get to the actual question that came in the chat. And that is, actually, I'll do the chat one first and mine will follow out of it. If Koppert was to commercialize Oscheius and the Heterorhabditis georgiana, will the Koppert product need to be applied in the billions because they'll lose their killer instinct? Or would they be producing them to our spec?
Yes.
Okay. Oop, we're losing you a little bit.
Okay, great.
If they produce the Oscheius and the HG, will they need to be applied in the billions?
So that is a great question. There is a potential that be doing lots of lab rats, and you take a lab rat out into the field and, you know, maybe it can't find food unless it's literally dropped in its food bowl. And then perhaps, you know, dropped in its food bowl with its mouth facing down on the food. So there's a chance that their mass propagation takes our particular species capacity to hunt and kill. So that's part of the quality control part. But ultimately, you know, what matters is it works. So if they apply their product at 1.5 billion per acre, and it gets us, say, 70% control, and then we apply our nematodes at, you know, only 12.5 million per acre, and we get 70% control. The point is, at the end of the day, you've got 70% control. So ultimately, what I care about is efficacy and a reasonable price. So I don't think growers care if they apply 12.5 million per acre or 1.5 billion. I think what they care about is, you know, how much it costs. Oh, they you know, got at the back end. Does that make sense?
Yep, that makes complete sense. So the follow-up question I had to that is there any, I know we've been doing work with the various people that you're doing these tries on, the various growers, to set their boom sprayers up to be able to distribute the nematodes without damaging the nematodes going through the nozzles. Is there any difference in making an application in the millions versus an application in the billions per acre in terms of sprayer logistics?
Right, no difference. No difference whatsoever. But it is still, like, I'm so glad, Allison, that you brought that up. It is so high, these nematodes at low pressure, and by taking off the nozzles or using the coarsest, crudest nozzle possible. Because one thing we have found is that when a nematode goes through a typical boom sprayer nozzle, it comes out the back end just destroyed. Just, I mean, killing insects after. And so, but that's easy. Like, whether you're applying 1.5 billion or 12.5 million, either way, you need to use low pressure and take off the nozzles. So.
And then,
yeah.
Awesome. I want to encourage people to continue to ask questions in the chat, as well as unmute and ask. I've got another couple of follow-up questions just to fill myself in on the side-by-side trials you're doing. Are these bed size trials, or are they split bed trials, or what's the setup of the head-to-head comparisons you're running?
Right. Great question. Two separate trials. The one that is head-to-head with the Koppert product is it's all within one bed, and these plots within the bed are about 100 feet long and about 5 feet wide. So these long, thin plots, 50 feet, which I feel is an adequate buffer. So, you know, nematodes do move, but they don't move very far. So buffer in those plots, 4 replicates of each treatment type. So I would say just very long, thin plots within a bed. The other trial is Koppert product, so Koppert versus control at a different marsh, and that one, it is bed comparisons, just because, you know, we had a lot of Koppert product, and so we just said to this grower, make sure that, you know, you randomly assign treatments to the beds that are getting the nematodes, and then can do that comparison, and we'll have data soon. Like that stuff, we will know by we should know by midsummer, because the killing happened last summer and fall.
Mhm.
The nematodes were applied late summer, obviously, and they did their thing. And so they were killing in late summer and fall, or perhaps, you know, putting insects to bed for winter with, you know, real sick. And so likely this spring, they will not emerge because they will have died. But we will be measuring that in the coming weeks. It's just that right now we're like 3 weeks behind. And yeah, we had that little flash of heat, but now we're back to cold and rainy, as I'm experiencing today.
It has indeed.
Yeah.
That's our awfully late spring. So, and then finally, are you going to be counting just sparg or sparg and cranberry fruitworm and flea beetle on those trial beds?
All three.
Awesome.
Yeah.
Awesome.
Yeah, I'm really hopeful. Like I am truly excited by this. I think this late summer timing, it makes sense. We just have to see what the data tell us. Yeah, and I can report on that. Well, should have data by, well, the flea beetle data won't be available until late summer, because you need to let the adults, it's the adult emergent that tells us how well we did. So probably for the winter cranberry meeting, I'll have all the data. Well, definitely by the winter cranberry meeting, I'll have the data in hand by then.
Awesome.
Yeah, I'll be happy to talk about it. Yeah.
I think we've gotten to our last 10 minutes of the call, so this is when everyone should ask their questions. Shawn, I guess thanks for this presentation so far, and we're happy to keep talking as long as questions keep coming in. But I want to thank Shawn with ARS for all of the work on the head-to-head comparison trials of the commercializable nematodes, as well as Alyssa Foss of DATCAP for the review of worker protection and safety, and then Suzanne Arendt of Red Forest Crop Consultants to give us our kind of grower updates for you know, our updates that we're seeing in the field as our IPM scouts make it out into the field. Go ahead and ask questions, folks. We've got a fair amount of people here, and we've got a lot of good brains on the call, so happy to hear what you're curious about. Let me see if I had any other questions. I had a question from Christelle to either the IPM consultants or any growers. What action thresholds are people using for sparg? Move us to the question slide. And I don't know whether.
Hi. Oh, sorry. Yeah. Who's up? Can you hear me?
Yes, Suzanne, it's you.
Oh, okay, great. So with the first generation of sparg, the literature states 2 to 3 larvae per sweep, but it's certainly, you know, more complex than that, because, you know, they'll web the upper leaves together, and sometimes you can't kick them out. So taking visual observations to include in that decision making is certainly critical as well. And then you know, I look at, you know, what is the history? Is this the first year I've been seeing them? If so, I usually try to push it a little further. What other insects am I seeing out there that could, you know, amplify the economic impact to the crop is another thing I think about. And then also, what's the grower's comfort level and what's the grower's budget? So sometimes, you know, even though I don't necessarily go with anything less than that, there's a lot of occasions where I will tolerate more based on sort of all those different factors.
Awesome. I know we have, you know, there's just kind of keeping track of the you know, where the publications are at and where actual grower use and grower decision making is at. So.
Oh yeah, I'll add one more thing. Sorry,
yes.
I just want to add one more thing. I also look at, you know, the larval stage. You know, you know, if the majority of them are first and second instars, certainly have a much better opportunity to manage that population. If they're near pupation, I just let them go.
That makes sense.
Thanks.
Absolutely, thank you. More questions from our participants?
Allison, this is Nick Hansen.
Hey, Nick.
Can we get feedback from people on the call? People that had hail, are you seeing increased amount of vegetative side shooting and plant injury versus people that did not have hail? Are they also seeing vegetative side shooting or not?
I am gonna ask for people to unmute themselves and talk while I set up a poll to ask this question in poll, but go ahead and respond verbally while I set that poll up. Great question.
This is Suzanne. So yes, I've definitely seen as the vines are starting to develop, especially in the warmer regions, those growers that did have hail that April 12th, or I'm not sure, that's the date that you're referring to. Yes, seeing vegetative side shoots, and I plan on doing a little bit more you know, documentation next week on, you know, what percentages have, you know, productive uprights alongside of those, you know, some areas, and it, you know, it doesn't matter what variety, obviously. But as they grow, we're going to continue to see what impact there may or may not be on the particular grower's yield. But you know, all of them that I've talked to where we are seeing those vegetative side shoots have made claims. And I am been told that insurance adjusters are going to be coming around in about 2 weeks. That way they'll have a better idea of, you know, the impact that that may be there.
Okay, thanks for answering that. While I got the poll set up, I am now ready to post that poll with, of course, really short sentences because I wanted to get it for you there quickly. But if you had hail, but you're not seeing side shoots, check that option. If you had hail and you are seeing side shoots, check that option. And if you were one of the lucky ones with no hail, there's an option for you as well. So far it's looking like most of our split is whether or not you had hail. So far, everyone who has responded, if they had hail, is indeed seeing side shoots, and that's certainly what I've seen in the field. We had a couple of yesterday at the station. Christelle was there and Jed was there, and Jed had brought 2 grad students, and we were looking to show buds and seeing indeed there are almost as many side shoots out here as we would have ever seen. I'm going to go ahead.
Allison?
Yep.
Did you give the option for no hail, but yet seeing side shoots?
I didn't give that option. I can end this poll and publish it and go add that option to ask again though, because that's a fair point.
That's the question.
Yep. Okay. All right. So.
And do we have a number of the percentage of cranberry producing area that actually got hit by hail anywhere? Has anybody have any thoughts on that?
I've heard kind of the informal polling results from the mini clinic. I haven't heard beyond that.
Yeah, I don't think at the mini clinic, people thought it was going to be too bad. I think we're going to be surprised with the damage we see. We're seeing reproductive uprights that are extending out and curling, where you can see the hail hit, the bud was healthy, but it did enough damage that not only did it damage the fruiting primordia that might have been in that on that side of the bud, it also must have damaged some of the cells that were going to eventually become the stem. And so if Amaya's on here, it'd be interesting to get some feedback from Amaya on any of her thoughts and what she's seen in the past on when we do have vegetative side shooting. Is there an impact on whether it'll still push that reproductive bud out as well? Right now, we're seeing that they are pushing out with some damage, but it's like Suzanne said, it'll probably be another week or two before we really understand the impact.
Yeah. I'm looking and I don't see Amaya on the call right now. I did post that new poll for hail inside shoots with all 4 possibilities present this time. The ones that I have seen, visually myself on marshes so far have been, I've not, I'm not. It seems like the side shoots are going to be purely vegetative, but like you said and Suzanne said, there might be a little bit of time before we'll have the final answer there. I've got 7 responses here, but I assume we'll be able to get a couple more. And also, if we have any other questions while people are filling in this poll, I guess we are to just 1 minute left. So I'll go ahead and display these results for you and the poll and share the results. So we did have either people had hail and do have side shoots, or people did not have hail and then don't have side shoots. So we right now it's looking like if you're seeing side shoots, it is likely attributable to that hail event that we had. And
there's also only 7 people that answered that, so
yeah. Yep. I do want to thank everybody for, I know sometimes it can be, if you're accessing this by phone, there's not a great way to do a poll. Like polls are pretty much only answerable by the growers who are on either a laptop or a full computer. So we have kind of that built-in handicap of not quite all the people will be able to answer. We are always happy to hear if you've got information and want to share it, go ahead and reach out to me either phone or email anytime. We're getting on to the 1 o'clock hour here, so I want to let people head off into their day and hope that Tom and Alex are having a good time checking on the new candidates for the communications position. And I'm sure we'll be hearing back on that shortly. I want to thank Shawn and Alyssa and Suzanne, as well as all of you guys for joining and for answering in our polls as we've had them. And I will be sending out information about June's mini clinic next. It'll be again the third Friday of June. It's going to feature Pam and Jed and Leslie. And I will again, if you see blunt-nosed leafhopper and you're willing to leave me 70 feet by 100 feet of an unsprayed plot so that I can do some pre and post bloom testing. I am all ears for that. So thank everyone so much, and I will we'll see you next time.